Hey, and thanks for clicking the link. Welcome to something we're trying new at ALS Canada for 2023 and for this year's research forum, which is a little bit different from what we've done in the past. In the past, we have sort of done a recap and wrap, rapid fire reiteration of the individuals and the content that was presented over the course of the 2 days of the scientific part. We're going to abandon that this year. We're going to try something a little different. So for historical context, the ALS Canada Research Forum for many years was a first come, first served by request agenda. So someone asked if they could give a talk and they would be slotted in. As the Canadian community evolved and the work that was being done became more broad, it really ended up being something that had to be curated a little bit more. So what we try to choose is people who haven't spoken in the recent year about their work, so more of an update. Individuals who have never spoken before or maybe new to the ALS community, so we can build collaborations and see what they're doing. And even young researchers to give them that platform at a conference to be able to explain their work and get those opportunities. So rather than do this afterward, we thought we would kind of explain the thoughts behind this year's agenda with the glance in advance. Now, hopefully you have also received what the actual written agenda is for you to be able to follow along and to be able to see as you go into the forum. And then this can be a supplemental piece for those who are wishing to learn a little bit more about what they're going to see. Now, I also hope that when you read this or you listen to this, you're as excited as I am, because this is how excited I am about the program this year. We've got a lot of great content highlighting amazing research from across the country. And if not, hopefully at least you're doing this and you're saying, yeah, not bad. I think I'm going to enjoy this and I'm definitely going to learn a few things. Now, if you're this is how you're feeling at the end of this, well, then I feel sorry. And we do welcome your feedback anytime. So please do reach out about feedback on forum programming moving forward, because we do ultimately want this to be the premier event for Canadian ALS research collaboration and sharing of ideas and really moving the science forward. Now, we're all going to be getting together on Saturday, May 6th for the Discovery and Impact Evening. It's going to be kicked off by Dr. Tim Miller from Washington University. Now, Tim is going to be hopefully talking a lot about tofersen. And as I talk about this right now, it is Monday, April 24th, the day before the PDUFA date for FDA to make a decision on tofersen. We are expecting some form of approval. I'm going to say that very boldly right now, and hopefully we'll be able to get some cool results explained during the talk. We're also probably going to see the genesis of how tofersen came about. We're going to be able to hear about how it has evolved trial design, including a biomarker that at the advisory committee for the FDA was really highlighted as a potential surrogate endpoint for clinical efficacy in ALS, neurofilament light, which is fantastic. And the groundbreaking, and I don't feel bad saying that, ATLAS trial, where people will be treated pre-symptomatically or are currently being treated pre-symptomatically in a randomized double-blind placebo-controlled trial incredibly innovative and new for the ALS field. But beyond that, Dr. Miller has worked on all kinds of pathogenesis of ALS over the years. He's still doing microRNA work, which hopefully will play into this. He's found some pretty cool potential therapeutic stuff with something called lovastatin recently. He does a lot of work on better understanding the clinical aspects of the disease, and through his work with the NEALS Consortium as a former co-chair, a big part of clinical trials beyond just tofersen. So we're really excited to have him, and he'll really kick it off with a bang. We're then going to go into a poster session. This is a great opportunity for the non-researchers in the audience and the researchers to spend some time together, for the non-researchers to see what the young researchers are doing across Canada, to get a good feel for how they communicate their research. And really, this evening is about a connection between the non-researcher stakeholders in the ALS community in Canada with the researchers. We're going to have some food after that, yeah, but then we're probably also going to have some tears. And one of the big parts of this evening has, for a very long time, been about trying to help the researchers see what it is they're working towards, and at the same time get that piece where the non-researchers get access to the research community. And the big one at the end of this is that we hope it fires everyone up and it gets those researchers ready to go back to the lab bench or back to the clinic and wanting to do everything they can to move the needle for ALS. Now, we then, after the Discovery and Impact evening, we end up going into 2 days of science. For those of you who are sticking around for those days, we're going to tell you a little bit about what what you're going to hear. For those who aren't sticking around, we're hopeful you'll get a little window into what the science party will be for the big nerd fest that we're going to have on the Sunday and Monday. Now, on the first day, it will be largely around translational or clinical research. We do this pragmatically, and we've done this for a number of years, and that's because even though there are a number of very great clinicians who stick around on the Monday, every year there are a few who struggle to be able to stay on the Monday of the research forum because they have clinical responsibilities. So we try to put the clinical aspects on the first day and dig into the fundamental pieces more on the Monday. So we're going to start on the first day at 8:30 with Dr. Sahara Karamula, who's a postdoctoral researcher with Dr. Yves de Koninck at Université Laval. She previously did a PhD with ALS research in the lab of Dr. Melanie Wooden at the University of Toronto, where she worked on chemogenetic mechanisms to slow down excessive firing that might be toxic in the motor cortex of ALS animal models. And the idea was that if it slowed this down, could it have a protective effect? Now, what she saw in her PhD work was that, in fact, it delayed the onset of an aggressive ALS mouse model called the G93A model. She's now continuing to do that work in Dr. de Konink's lab with actual small molecules or drugs. Which might be better for translating to the clinic. And I'm going to harken back a few years to when we had an opening speaker, Dr. Brian Kaspar, who at the time was using viral mechanisms to knock down or reduce SOD1. And he saw that one of these aggressive mouse models called the G93A model lived beyond 200 days with it. And he said that he always told people if he could get one of these to live beyond 200 days, he would retire. Now, from what I've seen from Sahara's work, I do believe she may actually have found a drug that could make them live beyond 200 days. And if I'm right on that, that's pretty amazing. But I'd also like to say, Sahara, please don't retire before you give the talk at the Research Forum on May 7th. So we'll be looking forward to that. We're then going to move forward to the CR Université de Montréal researcher, Dr. Alex Parker, who's pictured here with his postdoc, Dr. Audrey Labar. The two of them work with what's called C. elegans, a microscopic worm that actually can get motor neuron pathology and paralysis when given genes that confer ALS in humans. And using these models, what Audrey actually found several years ago was that probiotics could actually relieve some of those pathogenic mechanisms. And in fact, one of the lead candidates was something called HA114. Now, this brings us back to the idea that the microbiome could actually be connected into neurodegenerative diseases. And they've been doing follow-up work with Dr. Matthew Rees to actually see whether or not there's a metabolic signature, really trying to evolve out what the mechanism might be. All of this to say that, again, the microbiome or the gut bacteria and other living organisms might be connected to the brain and be able to have some influence. And what we're really excited to hear from this is also whether or not, or sorry, how HA114 is supposed to be moving forward into a clinical trial in the very near future. So details on that will be really, really exciting. We're then going to hear from Dr. Christopher Pearson at Sick Kids in Toronto. He's a world-renowned expansion mutation researcher and expert. This is him in his casual clothes, but this is him in his superhero clothes where he's looking at C9orf72. Now, a few years ago, he proposed that fluorodeoxyuridine was an opportunity to potentially therapeutically target C9orf72 by actually contracting the expansion mutations. We're hoping to hear a little bit about that, but he's also apparently got some really cool new data on better understanding the heterogeneity of C9orf72 disease, as well as potential other therapeutic options for C9. And then we're going to round out that session with Dr. Yasna Kriz from Université Laval. Now, she's a world-renowned ALS researcher on immune and inflammatory mechanisms in ALS. Traditionally, we've always looked at this as though these microglia turn into activated or toxic microglia that can affect motor neuron pathology. And one of the things that in recent years there's also been talk of is that it may also be important to be able to restore the protective and normal capabilities of these microglia. And in this whole realm, Dr. Kriz has been working on a molecular switch called SRSF3 that she's hoping to potentially move forward towards the clinic. And we're going to hear her latest results on that. We're going to go away for a break. We're going to come back to a clinical management session, which is typically what we have as the second session of the forum. It's going to be kicked off beautifully by two dynamic duo of Dr. Jocelyn Zwicker and Dr. Christine Watt, both working out of Ottawa and both palliative care experts. And they've been working on a really cool project to see whether or not earlier consultation, palliative care consultation in the clinical setting could actually improve quality of life as people progress with ALS. That's going to follow into a really great talk by a clinical research fellow working at Sunnybrook with Dr. Alessandro Abrahao and Dr. Lauren Zinman, and that's Dr. Andrea Parks. And she's actually looking at quality improvement in the way that we care for people with ALS, and particularly around psychosocial support for those individuals, and particularly around people who are younger, middle-aged individuals who are diagnosed with ALS, and what are the needs that they have? How can we learn from them better to better be able to provide that care moving forward? We're then going to hear from Dr. Gordon Jewett from the University of Calgary, also a clinical research fellow working with Dr. Lawrence Korngut and others. And he's going to tell us about his work with wearables. Now, not that wearables, obviously, but these kind of wearables. And the cool thing here is that for a long time, we've thought about wearables as a way to be able to potentially see the the functional decline of people living with ALS with the context of potentially using this as an outcome measure in clinical trials. That's still something that could happen as we learn more about this. But what Dr. Jewett's also doing is using this for better enhancement of care. So by using the information that's gleaned by these wearables, they'll be able to tell whether somebody needs to come to clinic earlier than their scheduled visit, or perhaps they can be scheduled a little bit later if they're not in need of that. And this way we could have a more tailored, personalized care regimen happening by using this kind of wearable technology. Really fantastic stuff. We're going to end that clinical management session with Dr. Lisiane Bouvier, who's a new assistant professor at McGill University. She just previously finished a postdoctoral fellowship with Dr. Yana Yunisova in the speech production lab at Sunnybrook. And she's a speech language pathologist who is now evolving alongside Dr. Yunisova AI techniques to better diagnose and understand the bulbar aspects of ALS, as well as in her acquired motor speech disorders lab, work with a relatively new outcome measure called an ALS bulbar dysfunction index, or BDI. Some really cool stuff coming out of that. And I think what's really also very cool about Lisiane's work is that she's adapting all of this as well to Quebec French. And obviously, as Canadians, we need to be able to make sure that these speech analyses that are associated with ALS are equitable across the country for both our national languages. We're going to have lunch, and we're going to come back to a translational panel that is moderated by Dr. Angela Genge, and it's going to be kicked off by Dr. Richard Robitaille. Richard's an expert in the neuromuscular junction, world-class expert, and when he came into the ALS field, one of the things he noticed was that little glial cells called perisynaptic Schwann cells that help to modify and modulate the neuromuscular junction where the motor neuron connects to the muscle, and keeping that healthy and making sure that that's maintained, these were abnormal in ALS models, and in fact were dysfunctional. And over many years, what he ended up finding was that a muscarinic receptor blocker called darafenacin was actually able to relieve that impairment in the PSCs, actually restore their function, and have a protective effect in ALS animal models. He's now been able to move this forward into clinical trial, and we're going to hear about what he's learned in taking his own work from the fundamental lab into the clinic. Speaking of that, we're going to have a couple guests who are going to come next who have done something similar. That's Dr. Emily Lowry, who is a co-founder, a senior advisor, and lead at Project ALS Therapeutics Core at Columbia, New York, as well as Aaron Fleming, who's the VP of Research and Development for Progenics. They've been working together for a number of years to advance a drug screening project looking at ER stress-related drugs that found a MAP4 kinase inhibitor in IPS models. You can find that paper online. And actually advance the work through to a first-in-human clinical trial of something called Procetin that is ongoing right now. So again, the learnings from taking something all the way from drug screening in ALS models and getting those into the actual clinic. And someone else who's done that really, really well is Dr. Steve Perrin, who is now leading Eledon Pharmaceuticals. Previously, you may know that he was the CEO and CSO for a long time of the ALS Therapy Development Institute, or ALS TDI. Now, TDI has been known for decades, in fact, for their ability to use ALS mouse models to actually screen for drugs. And they did a lot of work over the years trying to replicate what others had found as protective in these particular mouse models as something that could potentially be advanced to the clinic. And they weren't able to replicate a lot of those different things. But one of the things that they found that was very robust and replicative was an antibody against something called CD40 ligand. And so given that this is what the TDI research decided to hang their hat on, and that Steve then took to Analyxis Therapeutics, now to Eledon Pharmaceuticals, one has to be pretty excited about the potential of this particular therapeutic. Now, again, it's focusing on that immune and inflammatory modulation. If the biomarker data that I saw in terms of target engagement at the Niels, at the Neil Symposium. On their poster is anything to go by. It seems like it may be one of the most robust targeting that we've ever seen in this. It's actually now called Togobart, and when I say that, every time I hear Bob Probert, so maybe some other Canadians will think the same thing as me. I'm going to look at Zion, who you're going to hear from a little bit later, or hear about a little bit later. As a big hockey fan, hopefully same thing happened to him and I'm not crazy. Okay. We're going to have a break. We're going to come back. Dr. Max Senator, who is elected to the Order of Canada, the Royal Society, the Canadian Medical Hall of Fame. He's at UBC, and he's also done most of his work in the visual cortex, but recently he's worked a lot more on neurodegenerative diseases. In particular, he has been working on TDP-43 modulating drugs with primary peptides, and he's come to the forum to try to get some opinions on those particular strategies. So really do please engage with him. That will be really valuable as we see something from a Canadian company try to move towards the clinic. Speaking of trying to move towards the clinic, Dr. David Andrews at Sunnybrook has been collaborating with Amylyx to try to move anti-apoptotic inhibitors or inhibiting molecules towards the clinic. Now, some of you may say, if you've been in ALS for a long time, it was actually in the late 90s, early 2000s that anti-apoptotic mechanisms had their heyday in ALS, but none of those actually got moved forward really towards the clinic. And it might have been that the molecules we had to actually target them were not as advanced as the concept around whether apoptosis was an important feature in ALS. So they are trying to move that forward. That's going to be pretty exciting to hear about. And I'm personally glad, as a person who does these types of presentations, for the number of memes that Backs and Back will now give me. We're going to round out that session with Mark Shanuda, a PhD student with Dr. Janice Robertson at the University of Toronto. He's been doing a really great drug screening effort for a number of years. Including starting with computational methods, moving forward to primary neurons and other cell types, then moving into rodent models, really to hone it down into a drug that they want to move forward called JRMS. Now, before you ask me, where does JRMS come from? I think you can tell, Janice Roberts and Mark Shanuda. Now, I asked Mark, why wasn't it MSJR? And he said, well, don't you remember that time when Gary Armstrong's student, Christian Rampal, started working with Alex Parker on a drug, and it was a really great drug, but it was really poor marketing. Sorry for the joke. That's a really bad one. Were they going to bring something from the virtual forums we've run in the past, last year actually, called the Minute to Win It? These are little 1-minute advertisements for the posters that are out there, try to get people to their posters. But at the same time, we are going to once again have our great ALS Canada community ambassadors voting on these and giving away a poster prize to the people who provide the Minute to Win It. There will be another poster prize for the live posters and another thing coming up a little bit later. So lots of things to help nurture good engagement from the young researchers at the forum. We're then going to have a good long poster session for you to engage as attendees with all of the great researchers that are doing their poster presentations. Please do that. Let's make it a robust session, because after that, we're all going to get to go, if you're signed up, to the ping pong event, the networking event of the year. This is what we do at the ALS Canada Research Forum. It's very popular. Even if you're not planning to play ping pong and you're going, it's a great place for food, networking, have a drink, share your ideas, form collaborations, get some new friends, all that great stuff. And on the line for the ping pong tournament will be the Eisen Trophy. Now, the Eisen Trophy was named after Dr. Andy Eisen because of something called the Showdown we ran in 2021 at the Virtual Research Forum. This was pitting clinicians against fundamental researchers, where they were asked questions opposite to their expertise. And the ultimate winner was Dr. Geneviève Matt from the CR Schum, University of Montreal. And interestingly enough, the finalist that was against her was Dr. Christine Vandeveld from the same institution. Now, it didn't take long before they said, hey, we are pretty good as a team, as an institution. And they decided this year that they would put forward a challenge where UDM will challenge anyone else as a duo to a showdown 2023 version. And I will say we do have a competitor who will be revealed at the forum to challenge them. We have buzzers. It should be fun. 8:30 in the morning. Bring your breakfast in if you want to, but let's get us all warmed up for great science that day. We're also going to announce the poster award winners that morning. Make sure everyone's there, and we're going to get right into the science with Dr. Chantal Sefton from University of Laval. Now, Chantal's an expert on the biology of FUS from her mouse model of FUS, which I just wanted to highlight here because the fundamental researchers may not know. There's actually a phase 3 clinical trial going on right now by Ionis to knock down FUS, which will be really exciting and help us really advance our understanding of that biology. But when she gave her title, which is really cool, it was really cool that she was talking about early pathological changes in ALS and what she's learned and what she's going to share with us. I'm very, very excited to hear this talk. We're then going to hear from a recent Career Transition Award winner, Dr. Sylvia Posse, who is at University of Laval. She's a recent Canada Research Chair, which is fantastic. And she's been working on something called extracellular PPIA. Now, in people with ALS, this is seen elevated in the biofluids and also in models. And what she's found is that when this links up and binds to something called emprin, it actually activates NF-kappa B, activates MMP9, and actually leads to motor neuron death. And so what she's trying to do is prevent that interaction between the extracellular PPIA or cyclophilin A and the emprin by using an antibody technique that could go in and block that process from happening. We're then going to hear from another most recent ALS Canada Brain Canada Career Transition Award winner, and that is Dr. Philip McGoldrick, working with Dr. Janice Robertson at the University of Toronto. He works with something called nucleocytoplasmic transport. So that's actually the trafficking of things between the two major compartments of the cell. Really important stuff. Now, one of the things I like to highlight all the time is that even though this is a mechanism that's been connected to ALS for a number of years, the actual first connections of this really were coming from Canadian research, whether it was the GLI1 mutation discoveries by Dr. Patrick Dion and colleagues, or whether it was the isoform-specific C9orf72 antibodies that Dr. Janice Robertson had come up with, which showed that one of them actually linked to the nuclear membrane. These were postulated then to mean that nucleocytoplasmic transport was important in ALS, and it was just a few months later that a group of Americans all came out with different studies which showed this and connected it to gain-of-function aspects of C9orf72, so a toxic extra function that C9orf72 mutations conferred. Now, over the past several years, what Dr. McGoldrick's been doing is actually looking at nucleocytoplasmic transport defects with regard to a loss of function of C9orf72. Is the normal function of C9orf72 also involved in making sure there's proper trafficking between the nucleus and the cytoplasm? Now, all of this is really important for us to understand because given the Biogen trial that happened just last year and ended with a knockdown of sense-strand DPRs, target engagement shown that it approached one of the gain-of-function mechanisms and was actually leading to worse outcomes clinically, we need to better understand what's happening with C9orf72 biology to be able to move forward clinically and get to treatments as quickly as possible. We're then going to have a quick break. We're going to come back and we're going to hear from University of Alberta's Dr. Thomas Simon. Now, Thomas has never been to the forum before. He's looking for collaborators, so keep a good eye on this one. Speak to him after, speak to him before. Let's see what we can get going. He's an expert on the connections between the endoplasmic reticulum and mitochondria, as well as each of those individually. And he's done a review article last year or the year before, which is around MERCs, or mitochondrial endoplasmic reticulum contacts, and how this is affected in ALS. And he's also been working on certain things that are connected into ALS, such as TMX, which we've seen previously, that TMX can actually be involved in the ER stress that C9 DPRs can confer in ALS models. So there are connections there that will be really interesting. And if you see something that is of interest there, please do reach out and form collaborations with Dr. Simon. We're then going to hear from Dr. Renee Duville of the University of Winnipeg. Now, she's one of the pioneers of the idea of a reactivated retrovirus as contributing to the pathogenesis of ALS. And her work with Dr. Avi Nath at the NIH several years ago has led to the direction of her particular lab and how that might contribute to our understanding of ALS. Now, recently, she's been working on the idea that when these are reactivated, the ERV-K integrase actually causes DNA strand breaks that can't be properly repaired, and this may be conferring some of the motor neuron pathology that is seen. Now, one of the cool things that's happening right now that makes this very relevant is that, in fact, there is a anti-HIV therapy, Triumec, which is being tested in a very robust and long and large phase 3 clinical trial in Europe and Australia. The drug is called Triumec, and previously the Lighthouse 1 trial of this actually had some pretty intriguing results. One of the biomarkers coming out of this will be a reduction in HERV-K levels in biofluids, so hopefully hopefully this will help us get to the bottom of how we pursue this moving forward in ALS. We're then going to hear from Jay Ross, a PhD student with Dr. Patrick Dion and Dr. Guy Rouleau. Jay is a genetics expert, but he also does some really cool work on RNA biology, and in particular, methylation of RNA and how this can alter the RNA biology that's actually happening. These things called writers and readers and erasers, which are things like M6A, which can actually methylate the RNA and change its function. This is a process called epitranscriptomics, and it's not something that has been explored that heavily in ALS as of yet. So we're really excited to hear more about that. There was one paper last year from a collaboration with Dr. Sami Barmada, where they found, in fact, that the M6A methylation of the RNA is critical to TDP-43 binding and function. And what Jay's going to talk about is their C9orf72 specific work and how that complements what was found for TDP-43. We're then going to hear from Dr. Maria Vera Ugalde, who is an expert on understanding the life cycles of mRNA. And in particular, she's been collaborating at McGill with Dr. Heather Durham on the idea that the mRNA life cycle of HSP mRNAs might actually be conferring why motor neurons and ALS models are impaired in their ability to respond and create proper HSP responses. And if she can understand this life cycle well and be able to fix it, perhaps this breaks down the barriers that allows for HSP-inducing molecules or drugs to be able to be moved forward as potential therapeutics for the disease. Really exciting stuff, and glad to have her at the forum. We're then going to have a little break. We're going to come back. We're going to hear from a rapid fire set of talks from a bunch of great young researchers in the field, starting out with Zion Harji, who's working as a PhD student with Dr. Gary Armstrong at McGill University Montreal Neurological Institute. Bad joke alert. He's trying to create a betta fish, not a beta fish, but a better fish. And that is a zebrafish with a knock-in of TDP-43. So looking at more endogenous leveled expression. And then something that not a lot of people have ever done, if anyone, aging these out actually for over 3 years and actually seeing the neurodegenerative process happening in these fish. Really cool stuff, and looking forward to seeing that presented. We're then going to hear from Dr. Hanna Hakim, who's a postdoctoral researcher with Dr. Christine Vandevelde at the CRSHUM. And Dr. Vandevelde's been working for a number of years on a concept that when TDP-43 leaves the nucleus, this causes a loss of a really critical protein called G3BP1. And that is critical to stress granule function and all other aspects of RNA protein biology in these cells. And in fact, one of the things she's working on moving forward is how to use particular drugs to restore G3BP1 as a potential therapeutic for ALS. Now, what Dr. Fakim's going to do is actually try to understand those mechanisms by which TDP-43 may be misregulating the G3PP1 expression and all the complexities behind it needed to understand the process properly as it would be moved forward towards the clinic. We're then going to hear from Dr. Ulysses Corona Rodriguez, who's been working at the RCM with Dr. Marlene Offinger. He has actually been working on the complex biology of all the different structures of RNA proteins in the nucleus. It's not something that there's been a ton of data on, which is really exciting and quite complex. And in particular, he's looking at something with TDP-43 and FUS, and I think more so in this talk around TDP-43, and its connection to something called phosphoinositides and how that can regulate that important protein RNA biology happening in the nucleus in the ALS context. We're then going to hear from PhD student Miranda de St. Rome, working with Dr. Melanie Wooden at the University of Toronto. I spoke to you earlier about Dr. Katamula's work in terms of slowing down the firing of the motor cortex in the SOD1 context. What Miranda's doing is actually working in the same context, but in C9orf72 models, which is really exciting. And also leads us into the next talk, which is by Bile Geber-Gerges, working as a PhD student with Dr. Janice Robertson. And she had recently found that one of the normal functions of C9orf72, and in the context of a loss of function, there's actually abnormalities in synaptic glutamate receptor levels. And so what he's working on is what are the mechanisms in a C9 loss of function or knockout model of how this glutamate is misregulated, and how can this potentially tell us something about what, rescuing that by getting rid of that loss of function or regaining the normal functions of C9orf72. We're then going to finalize this whole session with I'm not sure if you got the doctor yet, Avi, but Avi Arthana-Day, working with Dr. Sanjay Kalra. She's got to be close to graduating soon. She's done amazing work. You can find her publication online. Working with Kalsnik data, longitudinal MRI data, as well as now we've got Capture ALS data coming forward, and published this really great paper, how you can see that motor cortex connectivity is reduced and dysfunctional in people living with ALS. And she's been able to coordinate not only this from the context of the imaging piece, but also through functional MRI and looking at chemicals such as N-aspartyl aspartate, which can allow us to be able to use this as a potential biomarker of that reduced motor cortex connectivity. Pretty cool stuff. We're going to have one last break. We're going to come back. We're going to have a session that's really focused more on the junior researchers. Now, I've written the word encourage up there. That's the symbol of the M&D Association UK's new initiative starting last year to really focus down and nurture the needs of early career researchers in the ALS M&D field. And we would love to be able to do something like this in Canada. In fact, on Saturday, May 6th, we're going to be gathering together all of the PhD students, postdocs, career transition award winners, and clinical fellows together to sort of ask them that question. What can we be doing moving forward in our strategic plan at ALS Canada to better have an initiative that supports early career researchers in their work moving forward? Now, as a little piece of that, what we're going to offer next is a unique thing. It's not that unique overall, a career panel, but this is going to be unique in that it's connected to the research forum. Everybody that's on the career panel will be somebody that was previously a young researcher sitting in the audience at our research forum in years past, going back to the early days of the forum, 2006, 7, 8. Dr. Katie Moyse was a PhD student with Dr. Michael Strong. She then went on to work for ABC News. She now is a professor at McMaster University working on scientific communications. So a really cool pathway that she'd be able to help tell people about. We're then going to hear from Dr. Gary Armstrong, who was a postdoctoral fellow working with Dr. Pierre Drapeau. He took the great academic path and is now a very successful assistant professor at McGill University Montreal Neurological Institute working on zebrafish. As you know, from earlier parts of this talk. We're then going to drag poor Colleen Doyle, who is the unsung hero of this whole forum, has pulled so much of it together. We're going to drag her up there at the very end on Monday because she will have really great insight coming from being a master's student with Dr. Elizabeth Meyering at the Research Forum for several years and becoming an employee of ALS Canada. So working in the nonprofit space now as our senior manager of research in CALS. We're then going to move into the clinical industry space with Christiana Salmon, who was a clinical research manager and clinical research coordinator at the Neuro with Dr. Angela Genge. She is now the director of clinical development for Curalis, so she can tell us about the clinical path for those in the audience that's relevant to. And then we're also going to hear from the fundamental path from Dr. Zachary Hawley, who was recently a PhD student with Dr. Michael Strong at Western in the audience of the Research Forum, and now a fundamental researcher and scientist at Biogen. We know that people have questions about all these different paths, so we thought it would be great to bring it from the perspective of people who would have been asking those same questions in the same audience years past. We're then going to wrap it all up. We're going to see how much you have retained. No recap and wrap. You're going to help us do it yourselves. And we're going to do something that we've never done before called the NERD, or Nerdy Elimination and Reward Dodgeball. So we're actually going to be asking questions about what you've heard over the course of the 2 days of the forum. And you're going to stay in by getting the answers right. Every time somebody gets an answer wrong, they're going to be said goodbye, and we're going to let them go for the end of the forum. Whoever's left standing will get a lovely Visa gift card for their engagement and the retention of all that they heard. That's a lot of stuff. I'm super excited about all this. We really would love for you to come, bring your questions, form some new collaborations. All of this discussion is what this research forum is about. It is why we put it forward at ALS Canada. It is to build a stronger ALS research community in Canada and to drive forward as our pieces towards the global effort on understanding and getting to a future without ALS. We can't wait to see you in this place, Toronto. Well, not this place, but this place. But you know what? It doesn't matter if it's out by the airport, because science rules.